Imagine, if you will, an invisible, incredibly sophisticated defense system operating within your body right now. It's a vast, highly coordinated network of specialized cells and organs working in perfect, silent harmony to tirelessly protect you from a constant barrage of threats you might not even be aware of.
It's truly remarkable when you think about it.
Today, we're taking an absolutely deep dive into the very foundation of this remarkable system, your immune system.
Yeah, getting right into the nuts and bolts.
Our goal is to go so incredibly deep, helping you master this information like an elite professional in the field, understanding not just what these components are, but how they intricately work together in a finely choreographed dance.
That's right. Our mission today is to truly unravel the intricate world of the immune system's cells, organs, and their unique microenvironments.
Okay.
We've poured over the comprehensive material you've shared, and we're going to peel back every single layer, exploring every topic and concept.
Really aiming for that mastery level.
We want you to walk away from this deep dive with a level of understanding that enables you to navigate this complex landscape with complete confidence.
And we're going to start right at the absolute beginning with the single remarkable cell that gives rise to all immune cells.
The ultimate ancestor cell, you could say.
Think of it as the ultimate progenitor. Then we'll trace their incredible journey through development in specialized primary organs, like the bone marrow and thymus, where they get their initial schooling.
Their basic training grounds.
From there, we'll see how these mature cells are strategically organized in secondary and even tertiary lymphoid tissues, forming dynamic command centers, ready to spring into action and mount a targeted, precise response.
It's a system of profound complexity, yet astonishing elegance, and we are going to unpack every single detail for you.
Okay, let's kick things off. Where does this story begin?
So let's open the curtain on the origin story of immunity with a truly fascinating cell, the hematopoietic stem cell, or HSC. This is where it all begins.
Okay, let's unpack this concept of a stem cell. When we talk about them, we're really highlighting two incredible, almost magical powers, aren't we?
Absolutely. Two defining characteristics.
First, their capacity for self-renewal. They can make more of themselves, ensuring a continuous supply.
Critical for maintaining the system long-term.
And second, their ability to differentiate, meaning they can transform into a diverse array of specialized cell types.
Becoming all the different blood and immune cells.
It's like having a master key that can unlock many different doors, leading to completely different cellular destinies.
Precisely. And it's crucial to grasp the distinction between different types of stem cells. Pluripotent embryonic stem cells, for instance, those found in a very early embryo or fertilized egg.
Right, the ones that can become literally anything.
Possess the astonishing capacity to generate an entire organism. They are, in essence, the ultimate biological blank slate. But the HSC, while incredibly versatile, is an example of a multipotent adult stem cell.
Okay, so more restricted.
This means it's tissue-specific. It can give rise to all the diverse cell types that make up the blood and immune system, from red blood cells to every type of white blood cell. But it won't suddenly turn into a liver cell or a neuron.
Got it. It stays within its lane, so to speak.
It's a highly specialized master key for our entire hematopoietic system.
And these HSCs, for all their power, are surprisingly rare, aren't they?
Incredibly so.
Our sources mention they primarily reside in the adult bone marrow, the spongy tissue inside our bones, though a very small number can also be found in the spleen and liver.
That's the main reservoir.
But here's where it gets really interesting. We're talking about a needle in a haystack. Less than one HSC is present per 50,000 cells in the bone marrow.
Yeah, think about that number. It's astonishing.
That's an astonishing rarity for such a foundational, life-sustaining cell. It makes you wonder how such a small number can maintain such a vast, dynamic system.
Their scarcity, in fact, isn't a limitation. It's a testament to how incredibly tightly regulated their numbers are.
Okay. Quality over quantity.
Think of it as a highly sophisticated biological accounting system. It's a delicate balance of controlled cell division, precisely timed cell death, and differentiation into mature cells.
All tightly controlled.
All of this is meticulously influenced by specific signals they receive within the specialized microenvironments or niches of those primary lymphoid organs.
That's the supportive neighborhoods.
Under steady state, homeostatic conditions, when the body is generally healthy and not under acute stress, most HSCs are actually quiescent.
Just kind of hanging out.
They're largely inactive, maintaining a small self-renewing pool, essentially waiting in the wings.
So when the body truly needs them, say during a severe infection where you're rapidly losing blood cells, or after intense medical interventions like chemotherapy that wipe out existing blood cells.
Then they kick into high gear.
They suddenly display an enormous proliferative capacity. You can see this strikingly demonstrated in mouse experiments. If you lethally irradiate a mouse, completely destroying its entire hematopoietic system,
A devastating situation for the mouse.
it would tragically succumb within about 10 days due to immune collapse and blood loss. But if you then infuse that mouse with fewer than 10,000 normal bone marrow cells from a donor,
A relatively tiny number of cells.
the entire system can be completely restored.
Yeah.
That, to me, is the immense, almost miraculous regenerative potential of these rare cells at work, rapidly repopulating the entire blood and immune system from just a tiny handful.
It's an incredible testament to their inherent power and the body's capacity for self-repair. Absolutely mind-boggling.
Indeed. And it underscores the historical challenge in immunology. Given their extreme rarity, how do you even identify and isolate these HSCs in the first place?
Exactly. For a long time, their only distinguishing feature was their function, their ability to create all blood cells when transplanted.
You couldn't just look at them and know.
No visual markers initially. This functional definition, rather than a visual or molecular one, led to some truly ingenious, almost puzzle-like process of elimination strategies.
This is exactly where classic experiment box 2.1 in our material truly shines, highlighting the ingenuity of early researchers. They used a method called panning.
Panning, right, a clever approach.
The core idea was brilliant. They reasoned that undifferentiated HSCs, by definition, wouldn't express the lineage markers or lin plus markers that are found on mature differentiated cells.
The logic being, if it's mature, it's not a stem cell.
So they incubated a sample of mixed bone marrow cells with specific antibodies against these lin plus markers. And these antibodies were bound to plastic dishes.
Sticking the mature cells down.
The mature cells with their lin plus markers would literally stick to the plastic, while the lin cells, which were enriched with HSCs, could then be gently dislodged and collected.
Leaving the good stuff behind, relatively speaking.
This initial negative selection method remarkably achieved a several thousand-fold enrichment, giving them a much more concentrated pool of potential stem cells to work with.
It was a groundbreaking step in isolating such a rare cell type, a huge leap forward.
And then, the scientific landscape was utterly transformed by two technological revolutions: the advent of monoclonal antibodies and the development of flow cytometry.
Game changers, truly.
Monoclonal antibodies were a game changer because they allowed for highly specific targeting of individual proteins on cell surfaces, far more precisely than previous methods.
Super specific tags.
And flow cytometry, a powerful machine directly inspired by fluorescence microscopy, could rapidly identify, separate, and even recover individual cells based on their unique protein expression patterns by using fluorescent reagents.
Imagine you could put 1 million cells into a machine and it could instantly sort them based on which tiny glowy tags they carried, sorting at incredible speed.
So pioneers like Irv Weissman and his colleagues, recognizing this potential, masterfully combined these techniques. They moved beyond just negative selection to incorporate positive selection.
Looking for what is there, not just what's missing.
They looked for cells that were lin, meaning they didn't have mature lineage markers, but also expressed specific newly discovered markers like CD34, SK1, and KICKIT.
Adding more layers of identification.
These became known as LSK cells. LinSCAL1 plus CKIT plus dairy. What's fascinating is that even this highly enriched population, which still represents less than 1% of total bone marrow cells, is functionally heterogeneous.
Still not perfectly pure.
It still requires even more markers, a deeper dive into their surface profile, to fully sort out and pinpoint the true long-term HSCs.
It shows the incredible hidden complexity even within a supposedly pure population. We're always finding more layers.
And to measure the precise success of this enrichment, they reverted to that very elegant functional assay, transplanting the isolated cells into lethally irradiated mice.
Back to the ultimate test. Can it rebuild the system?
If you could restore a mouse's hematopoietic system with significantly fewer cells than an unpurified bone marrow sample, you knew you had effectively enriched for HSCs.
Right. The fewer cells needed, the better the enrichment.
For example, achieving a 1,000-fold enrichment meant you needed 8,000 times fewer purified cells to achieve the same life-saving result.
This isn't just about counting cells. It's about proving their functional potency in the living system. A really robust measure.
And within these LSK-HSCs, there's even more fascinating nuance, our almost hierarchical structure.
Yeah, it's not just one type of HSC.
We now recognize subpopulations with different self-renewal capacities and proliferative potentials. At the top are the long-term HSCs, or LT-HSCs,
The true long haulers?
which are the most quiescent, meaning they divide infrequently, but they retain their full multipotent capacity and can sustain hematopoiesis throughout the organism's entire life.
The ultimate reservoir.
Then we have short-term HSCs, or ST-HSCs, which divide more frequently but have a more limited self-renewal capacity.
More active, but finite potential.
And finally, the rapidly proliferating multipotent progenitors, or MPPs. These MPPs have very limited self-renewal, but retain the crucial ability to give rise to both lymphoid and myeloid lineages.
Ready to commit, essentially.
It's also worth noting that PKIT, one of those key markers we mentioned, is actually a receptor for the cytokine SCF, a signaling molecule that directly promotes MPP development.
So even at this very early stage, these cells are already receiving precise molecular instructions telling them what their next step should be. They're listening to their environment.
This brings us to a crucial fork in the road, the moment of lineage commitment.
A point of no return, really.
As HSCs progressively differentiate, moving from LT-HSC to ST-HSC and then to MPP, they ultimately shed some of their original versatility, losing their ability to self-renew indefinitely.
Becoming more specialized.
At the MDP stage, they make one of two fundamental and irreversible lineage choices. They either commit to becoming a myeloid progenitor cell, often termed a common myeloid progenitor or CMP, or a lymphoid progenitor cell, known as a common lymphoid progenitor or CLP.
Path 2. This decision sets the stage for their entire future function.
So on one path, the myeloid progenitor, the CMP, gives rise to our immediate responders, the cells of the innate immune system.
The first line of defense.
This includes red blood cells for oxygen transport, platelets for clotting, and a whole host of white blood cells. Granulocytes like neutrophils, eosinophils, and basophils, as well as monocytes, macrophages, and some types of dendritic cells.
A very diverse group.
These are the rapid response team, the first cells on the scene when an infection or injury strikes, providing immediate, broad-spectrum defense.
In contrast, the lymphoid progenitor, the CLP, takes a different path, giving rise to the adaptive immune cells.
The specialized forces.
These are the highly specialized intelligent forces, B and T lymphocytes, innate lymphoid cells, ILCs, and specific antigen-presenting dendritic cell populations.
The memory makers.
These are the cells that generate a highly refined antigen-specific response, crucially building immune memory so that future encounters with the same pathogen are met with a faster, more potent defense.
That's key.
What's absolutely vital to understand here is that as cells commit down either the myeloid or lymphoid path, they progressively lose their ability to self-renew and to contribute to other lineages.
They become true specialists.
This ensures that their function becomes increasingly specialized and efficient. No jack of all trades here.
We can trace this progressive loss of potential with a fascinating specific example, the B-cell development pathway. Think of it as a series of narrowing gates.
A great analogy.
MPPs, those multipotent progenitors, start by expressing a receptor called LAT3, and at this point, they transform into lymphoid-primed multipotent progenitors, or LMPPs.
LMPPs, okay.
What's significant here is that even at this early stage, they've already lost the potential to become erythrocytes or platelets. They're now firmly on a lymphoid-leaning track.
Already closing some doors.
And the journey continues. As they commit further down the lymphoid path, levels of stem cell antigens like c-KIT and SCA1 begin to fall, signaling their decreasing stem cell-like properties.
Shedding their stemness.
Simultaneously, they start expressing genes for RAG12 and TDT, which are crucial enzymes involved in generating the diverse receptors that lymphocytes will eventually use to recognize antigens.
Getting the machinery ready for adaptive immunity.
This expression marks them as early lymphoid progenitors, or ELPs. Some of these ELPs will then migrate out of the bone marrow to a different primary lymphoid organ, the thymus, to begin their journey towards becoming T-cell progenitors.
Off to T-cell school.
The rest, however, remain in the bone marrow, continuing their development as B-cell progenitors. As they do so, their interleukin-7 receptor, IL-7R levels, dramatically increase, transforming them into common lymphoid progenitors.
The CLPs.
The CLPs, right? Which are now c-KIT, SCA1, IL-7R plus SUMA. By this stage, they've lost all myeloid potential, but still retain the incredible ability to mature into any lymphocyte lineage. a T cell, a B cell, or an ILC.
Still flexible within the lymphoid world, it's a beautifully orchestrated journey of successive commitment and specialization.
And this entire journey, this intricate ballet of differentiation and commitment, is orchestrated at a fundamental genetic level. It's not random.
Not at all. It's highly programmed.
HSCs actually maintain a relatively large number of genes in what we call a primed state. Think of it like keeping the garage door open on potential pathways.
Keeping options open, genetically speaking.
Making those genes accessible to the transcriptional machinery, ready to be turned on. Then, environmental signals within those specialized niches come into play.
The local instructions matter.
These signals upregulate specific transcription factors. These are like molecular master switches, proteins that bind to DNA and activate or repress gene expression, driving the cell down one particular developmental pathway.
Flipping the right switches.
Crucially, as the cell commits, the chromatin regions containing genes not needed for that chosen pathway are actively shut down, becoming inaccessible.
Locking down the unused pathways. This ensures efficiency and prevents unwanted gene expression.
Figure 2.2 in our source beautifully illustrates some of these key genetic regulators. For example, when we consider HSC quiescence, proliferation, and differentiation, we see a complex interplay of factors like GATA2, RUNX1, SOLTAL1, PU.1, ERG, and many others.
It's a whole symphony of gene expression controlling the basics.
But then, there are specific factors that decisively regulate the myeloid versus lymphoid lineage choices.
The real decision makers.
For instance, Icaros is absolutely required for lymphoid development. Without it, animals are severely immunocompromised, highlighting its essential role.
A critical lymphoid factor.
PU.1 is particularly fascinating because its concentration acts as a cellular dial. Low levels favor lymphoid differentiation, while high levels push cells firmly towards a myeloid fate.
Like a dimmer switch for lineage choice.
And Notch1 is a critical signaling pathway that specifically induces lymphoid progenitors to commit to T cell development rather than B cells,
pushing them towards the thymus path.
even within the myeloid lineage. Specific factors like GATA1 direct erythroid development over granulocyte-monocyte lineages, and PU.1 also influences that choice.
Fine-tuning within the major branches, too. It's truly a sophisticated, multi-layered regulatory network, a marvel of genetic programming.
So we've talked about where these cells come from and how they commit to different paths, but how do we, as scientists, actually tell all these incredibly similar-looking cells apart?
Yeah, that's a fundamental challenge, especially early on.
Historically, before the age of advanced molecular biology, it was all about what you could see under a microscope, often with the help of specialized dyes.
Morphology and staining were key.
Microscopists would use pH-sensitive stains like hematoxylin, which avidly binds to basophilic nucleic acids and stains them a deep blue, and eosin, which binds to eosinophilic proteins in cytoplasmic granules and stains them vibrant pink.
H&E staining, the classic.
This allowed them to distinguish, say, a lymphocyte with its dense, round nucleus from a granulocyte, with its segmented nucleus and distinct cytoplasmic granules.
Making incredibly astute inferences about cell type and even function just from these visual cues, a real art form.
But then the field was revolutionized. Fluorescence microscopy greatly enhanced our ability to identify molecular details within cells by tagging specific components with glowing molecules.
Adding molecular specificity to the visual.
And in the 1980s, this technology directly inspired the development of the flow cytometer. This invention truly was a paradigm shift in immunology.
Absolutely changed the game.
It allowed us to rapidly and quantitatively measure the presence of multiple surface and internal proteins on individual cells, one by one, at speeds of thousands of cells per second, a level of detail and throughput previously unimaginable.
Imagine sending cells through a laser beam, and each one flashes a unique molecular barcode. High-throughput analysis.
And this is where the cluster of differentiation, or CD nomenclature, comes in.
Bringing order to the chaos.
Before this, different labs would discover the same surface protein on an immune cell and give it a different name, making communication within the scientific community a chaotic mess.
Total confusion. Everyone had their own names.
So in 1982, an international group of immunologists got together and standardized the identification of these surface proteins, giving them a universal CD designation.
A much-needed universal language.
Think of it like a global identification tag for immune cells. For instance, what was once called Thy1 is now universally known as CD90, and B71, B72 are now CD80, CD86.
This system brought clarity and allowed researchers worldwide to compare results accurately. Essential for progress.
Indeed, these technologies, coupled with our increasing ability to precisely edit animal and cell genomes, have collectively revealed an unanticipated and still incomplete diversity of hematopoietic cell types, functions, and interactions.
We're still discovering new nuances. The picture keeps getting richer and more complex.
Table 2.1 in your material provides a practical snapshot, listing key data like the number of cells per cubic millimeter in the blood, their total leukocyte percentages, and their average lifespans for major myeloid and lymphoid types.
A useful reference point.
What's truly striking when you look at that table is the immense variability, especially in lifespan. For example, neutrophils might only live for a few days in tissue, constantly being replaced.
Very short-lived workhorses.
Whereas some memory cells can persist for decades.
Holding on to that immunological memory for a lifetime. This underscores the incredibly dynamic and adaptive nature of this system, constantly churning and renewing to meet the body's demands.
Now that we've delved into where these incredible cells originate and how we identify them, let's turn our attention to the myeloid lineage, which truly represents the innate front line of defense.
Right, the innate arm. These are our immediate responders, the rapid action forces of the immune system.
They're the first ones in.
That's right. The myeloid lineage encompasses all red blood cells, platelets, and the white blood cells of the innate immune system. Granulocytes, monocytes, macrophages, and specific dendritic cells.
Quite a crew.
They're designed for speed and broad-spectrum action, to respond rapidly and non-specifically to pathogen invasion, and crucially to communicate that initial threat to the more specialized adaptive immune cells.
Bridging the gap. Okay, first up are the granulocytes. These are often the very first responders to an infection.
Often the first white blood cells on the scene.
You can spot them visually by their distinctive multi-lode nuclei, which look quite different from the typical round nuclei of lymphocytes.
That segmented nucleus is a key feature.
And their most defining feature are those cytoplasmic granules, membrane-bound vesicles, packed with a potent arsenal of proteins.
Hence the name granulocytes.
These granules aren't just for show. When released, their contents directly damage pathogens, regulate the activity of other white blood cells, and even contribute to tissue remodeling at the site of inflammation.
A real chemical warfare toolkit inside each cell. Table 2.2 in our material gives you a sense of just how diverse these granule contents are, including everything from powerful proteases that break down proteins to antimicrobial defensins that directly kill bacteria.
A lot packed into those tiny sacs. Among granulocytes, neutrophils are by far the most numerous circulating leukocytes, typically making up a massive 50% to 70% of all white blood cells in your bloodstream.
The most abundant type. The foot soldiers in many ways.
But despite their abundance, they have a surprisingly short circulation time, only about 7-10 hours in the blood, and an even shorter tissue lifespan, typically just a few days.
Constantly being produced and replaced. High turnover.
This means they are constantly produced and replenished. A transient increase in circulating neutrophils, known medically as leukocytosis, is actually one of the most common and immediate indicators of an ongoing bacterial infection.
A classic sign doctors look for in blood tests.
A clear sign of their rapid deployment. They swarm to infection sites, efficiently phagocytose and engulf bacteria, and secrete a battery of antimicrobial and tissue remodeling proteins.
Eating the invaders and releasing weapons.
They are, in fact, the main cellular components of what we commonly call pus.
Yeah, pus is mostly dead neutrophils after the battle.
Which is essentially a collection of dead neutrophils and cellular debris. While they were once considered disposable cells, merely foot soldiers of the immune system, we now understand they play a much more sophisticated regulatory role, actively shaping the subsequent adaptive immune response.
Right. They're not just killers. They talk to other immune cells and influence the longer-term response.
Then we have eosinophils, which are instantly recognizable under a microscope by their brilliant, almost fluorescent pink granules when stained with H&E.
That bright pink color is unmistakable. Eosinophilic equals eosin-loving.
Their primary and vital role is in our defense against multicellular parasitic organisms, particularly helminths or parasitic worms.
Really important for tackling larger parasites.
When these invaders are detected, eosinophils cluster around them, releasing destructive granule contents like eosinophil peroxidase, EPO, and major basic protein, MBP, to damage the parasite's membranes.
Trying to dissolve the worm from the outside, essentially.
They are highly motile and found in abundance in tissues exposed to the external environment, like the small intestines. But interestingly, in regions of the world where parasitic infections are less prevalent,
like in many Western countries,
the eosinophils are perhaps better known for their dual role. They are major contributors to the symptoms of asthma and allergies, mediating inflammation.
Yeah, they're heavily involved in allergic reactions, a double-edged sword.
And perhaps surprisingly, they also secrete cytokines that can directly regulate the activity of B and T lymphocytes, showcasing their broader immune influence.
Again, showing more complex roles than initially thought. They're communicators, too.
Basophils are next, and they are relatively rare in the circulation, but don't let their scarcity fool you. They are potent responders.
Very low numbers, but pack a punch.
They are characterized by large, distinctive basophilic granules that stain a deep blue. Like eosinophils, they are involved in the response to helminths.
Also part of the anti-parasite team.
But their most impactful role might be their rapid release of powerful compounds, most notably histamine, when they bind to circulating antibody-antigen complexes.
Histamine is the big one.
Histamine dramatically increases blood vessel permeability and smooth muscle activity, allowing other crucial immune cells to more easily access the infection site.
Opening the gates for reinforcements.
They also recruit other immune cells, including eosinophils and lymphocytes, into the fray. And yes, it is the release of histamine by basophils and mast cells that causes many of the familiar and uncomfortable symptoms of allergic reactions.
Absolutely. That runny nose, itching, swelling, often histamine-driven.
And closely related, with similar roles in combating parasitic worms and contributing to allergies, are mast cells.
Often discussed alongside basophils.
What's particularly unique about mast cells is their maturation process. They are released from the bone marrow as undifferentiated precursors and only mature after they leave the blood and settle terminally in various peripheral tissues.
They finish growing up in the tissues, not in the marrow or blood.
Like the skin, connective tissue surrounding blood vessels, and mucosal epithelial tissues lining our respiratory and digestive tracts.
Stationed strategically at the body's interfaces.
Once matured, their granules, much like basophils, are loaded with histamine and other pharmacologically active substances ready for rapid release.
Also packed with histamine and other potent mediators.
While they share many functional similarities with basophils, recent research, diving into their developmental origins, increasingly suggests they have distinct lineages and specialized functions.
Yeah, the thinking now is they are related but distinct, not just tissue-resident basophils. Adds another layer of complexity.
Always more complexity. Moving beyond the granulocytes, we come to the myeloid antigen-presenting cells, or APCs.
Ah, the crucial communicators.
This critical group includes monocytes, macrophages, and dendritic cells, all of which function as professional APCs, or PAPCs.
Professional antigen presenters, their main job is showing antigens to T cells.
They are the essential cellular bridges between the rapid, nonspecific innate immune system and the highly specific adaptive immune system.
Connecting the two arms of immunity.
Upon making contact with a pathogen, they become activated and communicate the presence of that infection directly to T lymphocytes in the lymph nodes by processing pathogen-derived proteins and displaying their peptide fragments on specialized cell surface molecules called MHC class II molecules.
That presentation on MHC class II is key for activating helper T cells.
And when these professional APCs get activated, they undertake three major coordinated activities that are absolutely essential for kickstarting an adaptive immune response.
A three-pronged activation strategy.
First, they secrete a symphony of signaling proteins, cytokines and chemokines, which act as molecular alarms, and GPS signals to attract and activate other immune cells to the site of infection and to the lymph nodes.
Sounding the alarm and calling for backup.
Second, they efficiently internalize pathogens through processes like phagocytosis, digest their proteins into small peptide fragments, and then crucially display these peptides on their cell surface via those MHC class II molecules we just mentioned.
Chop up the enemy and display the pieces.
And third, they upregulate the expression of what we call costimulatory molecules. These are like critical handshake signals absolutely required for optimal activation of helper T cells.
The second signal. Without it, T cells might ignore the antigen or even become unresponsive. Very important.
Without them, the T cells won't fully engage and activate.
Exactly. Prevents accidental activation.
Monocytes, typically comprising 2–12% of your white blood cells, are a fascinatingly heterogeneous group.
Not just one type of monocyte.
They are essentially circulating precursors that migrate into tissues and then differentiate into a diverse array of tissue-resident phagocytic cells.
The seeds for tissue macrophages and some DCs.
Modern research has further categorized them into distinct populations. Inflammatory monocytes, which are the rapid response subset, quickly entering inflamed tissues during an infection to fight pathogens.
Rushing into the fire.
And then there are patrolling monocytes, which, in contrast, slowly crawl along blood vessel walls, constantly monitoring for signs of damage or infection.
Like sentries pacing the walls.
And can even act as a reservoir for resident monocytes in the absence of acute inflammation.
A fascinating population constantly surveilling the vasculature.
And when those monocytes migrate into tissues, particularly during an infection, they can differentiate into macrophages.
The big eaters.
These inflammatory macrophages are truly expert phagocytes, incredibly efficient at engulfing and clearing not only pathogens, but also damaged host cells and cellular debris, like the cleanup crew.
Hoovering up the mess and the bad guys.
They have a critical dual role, directly clearing pathogens and acting as professional HSCs for T lymphocytes, presenting those processed antigens.
Killing and telling, basically.
What's genuinely surprising, and a more recent scientific breakthrough, is that most tissue-resident macrophages, like the Kupffer cells in your liver, microglia in your brain, or alveolar macrophages in your lungs, actually arise very early in life from embryonic cells.
Yeah, this was a huge shift in thinking. They're not all derived from adult monocytes.
Not solely from circulating monocytes. They are long-lived, self-renew, and form a committed, permanent part of the tissue microenvironment, each with specialized tissue-specific functions, as highlighted in Table 2.3.
They set up shop early and stay there, doing unique jobs depending on the tissue. This fundamentally changes our understanding of tissue immunity.
And their phagocytic efficiency can be dramatically enhanced by a process called opsonization.
Making things tastier for phagocytes.
Macrophages and other phagocytes express specialized receptors for certain classes of antibodies. If a pathogen is coated with antibodies, which effectively act as opsonins, derived from the Greek word meaning to supply food or make tasty, this coating acts as a molecular flag.
The antibody tags the pathogen for destruction.
This binding can astonishingly increase the rate of phagocytosis by up to 4,000-fold in some studies, making the pathogen far more palatable, so to speak, for the macrophage to engulf and eliminate.
4,000 times. That's a massive boost. It's a prime example of how the innate and adaptive systems cooperate for enhanced defense.
Here's where it's really interesting. Dendritic cells, or DCs. The late Ralph Steinman won the Nobel Prize in 2011 for their discovery in the mid-1970s.
A well-deserved Nobel. Their discovery truly reshaped immunology.
Which completely reshaped our understanding of immune initiation. These cells are truly critical for kickstarting the adaptive immune response.
They are often considered the most potent APCs for activating naive T cells, the master initiators.
They're named for their nerve-like dendrite extensions, which dramatically increase their surface area, allowing them to efficiently browse and interact with lymphocytes.
Those extensions maximize contact with T cells.
What's also become clear is their surprising diversity. They are more varied than once thought, arising from both myeloid and lymphoid lineages, and scientists are still working to fully clarify their distinct functional roles.
It's a complex family of cells, not just one type. Still much to learn.
Their key strategic trick is performing distinct functions in different anatomical locations. Immature DCs, residing in peripheral tissues like your skin and mucous membranes, act as vigilant sentinels.
Patrolling the borders.
Their primary job is antigen capture, which they do with remarkable efficiency through phagocytosis, receptor-mediated endocytosis, and even pinocytosis, where they can imbibe fluid volumes of 1,000 to 1,500 cubic millimeters per hour.
Constantly sampling their environment, literally drinking it in.
After they encounter and capture an antigen, a sign of infection, they undergo a crucial maturation process.
They get the signal and change their behavior.
During maturation, they lose their capacity for large-scale phagocytosis and pinocytosis, but become significantly better at antigen presentation.
Stop eating, start talking.
They also upregulate those crucial co-stimulatory molecules that we mentioned earlier, which are absolutely essential for providing the second signal to activate naive T cells.
Getting fully equipped to activate T cells properly.
Once matured, they embark on a journey, migrating from the periphery to the lymphoid organs, specifically the lymph nodes, to present the captured antigen to circulating T cells and initiate that targeted adaptive response.
They travel to the command centers to report the invasion.
It's important to clarify, though, that not all cells with dendrite-like extensions are the same. For instance, follicular dendritic cells, or FDCs, are distinct.
Right, a potential point of confusion. FDCs are different beasts.
Crucially, they do not arise from hematopoietic stem cells, and they are not professional antigen-presenting cells for T cells.
Different origin, different job.
Instead, FDCs reside exclusively within the B-cell-rich follicles of secondary lymphoid tissues, and their primary role is to regulate B-cell activation by presenting unprocessed antigen directly to them.
They present whole antigens to B cells, not processed peptides to T cells. This distinction underscores the incredible specialization and complexity within the immune system.
Okay, rounding out the myeloid derivatives.
Beyond the immune cells, the myeloid lineage also gives rise to two other absolutely crucial blood components that are vital for our survival. Red blood cells, also known as erythrocytes, and platelets.
Oxygen carriers and clot formers.
Erythrocytes arise from myeloid progenitors in the bone marrow and are primarily responsible for delivering oxygen to every tissue in our bodies, a job they perform thanks to their remarkably high concentration of hemoglobin.
Packed full of hemoglobin.
Uniquely in mammals, mature red blood cells are anuclear, meaning their nucleated precursors, called erythroblasts, actually extrude their nuclei in the bone marrow before entering circulation.
They kick out their nucleus. Why?
This allows them to pack in more hemoglobin and adopt that characteristic biconcave shape, which is optimized for squeezing through capillaries and maximizing oxygen exchange. More room for cargo.
But don't let their primary job of oxygen transport fool you. Red blood cells also play a direct and increasingly recognized role in immunity.
More hidden talents.
They can release signals that contribute to innate immunity, express antibody receptors that help clear harmful immune complexes from the circulation, and even generate antimicrobial compounds like nitric oxide or NO.
So they're far from mere oxygen carriers. They're quiet contributors to our defense. Still much to learn there, too.
And then there are megakaryocytes. These are truly massive myeloid cells residing in the bone marrow.
Giants of the bone marrow.
And they're the factories that literally give rise to thousands of platelets.
They fragment themselves into tiny platelets.
Platelets themselves are tiny nuclear cell fragments that circulate in the blood, and they are absolutely crucial for blood clot formation.
Essential for stopping bleeding.
Clots do far more than just prevent blood loss at a wound site. When they form at epithelial barriers, they provide a vital physical barrier against pathogen invasion, essentially sealing off potential entry points.
Walling off the breach. Like mature red blood cells, platelets remarkably don't have nuclei.
Okay, that covers the myeloid side in incredible detail.
Yeah.
Now let's shift our focus to the lymphoid lineage. These are the true specialists, the architects of our adaptive immune response.
Lymphocytes, which broadly include B cells, T cells, and innate lymphoid cells, ILCs, are indeed the principal cellular players in our adaptive immune response and the ultimate source of long-lasting immune memory.
The smart bombs and the memory banks.
They constitute a significant portion of our white blood cells, making up 20–40% of circulating leukocytes and an astounding 99% of cells found in the lymph.
Dominant in the lymph fluid.
Think about that. Roughly 1,012,000,000 lymphocytes are continuously circulating throughout our bodies, constantly patrolling, ready for action.
1 trillion. That's an astronomical number.
Microscopically, these cells are quite distinctive. Small, round, and largely dominated by their nucleus.
Big nucleus. Not much cytoplasm usually.
What's particularly interesting from a diagnostic perspective is that mature B and T cells actually look identical under a standard microscope.
You can't tell them apart just by looking.
Nope. This is precisely why we rely so heavily on their unique surface protein profiles to tell them apart, using that standardized CD nomenclature we discussed earlier.
Back to the CD markers.
Table 2.4 in your material conveniently lists some of those common CD markers, like CD3 being a universal marker for T cells, or CD19 and CD20 for B cells, that allow us to distinguish these critical populations.
Molecular identification is key.
But the true genius of B and T cells lies in their antigen-specific receptors, the B cell receptor, BCR, and T cell receptor, TCR, respectively. These receptors are unique to each cell.
The source of their specificity.
What's astonishing is the sheer diversity they can generate. Over 1 billion different specificities are possible within an individual.
1 billion. How is that even possible?
Through an amazing process of gene rearrangement we'll cover later. Yet every single antigen-specific receptor on an individual cell surface is identical in structure and specificity.
So one cell, one target.
Exactly. This means one B cell, for example, will only recognize one specific molecular pattern, and here's the magic. When that specific T or B cell encounters its corresponding antigen, it rapidly divides.
Clonal expansion.
And all its progeny form a clone with that exact same receptor specificity. This process, known as clonal expansion, allows the immune system to generate a massive, highly targeted army from just a few specific soldiers.
Amplifying the right response. That's right, clonal expansion in action, magnifying a tiny specific response into a powerful one. Newly formed B and T cells are considered naive until they encounter their specific antigen for the very first time.
Fresh out of school, haven't seen action yet.
Upon that initial contact, they undergo a rapid transformation. They proliferate extensively and differentiate into effector cells, which are the short-lived, highly specialized fighters that carry out the immediate battle.
The soldiers for the current fight.
But critically, they also differentiate into memory cells, which are long-lived, persist in the host, and are ready to respond much faster and far more efficiently if that same antigen is ever encountered again.
The veterans, remembering the enemy, this is the fundamental basis of primary versus secondary immune responses, and it's why vaccines work.
So let's delve into B lymphocytes, our powerful antibody factories.
The antibody producers. The B in their name originally comes from the bursa of Fabricius in birds, a specialized lymphoid organ where B cells mature.
The bird connection.
But for mammals, including humans, the name is equally apt because the bone marrow is their major site of maturation.
Bone marrow for B cells in us.
Their defining feature, the molecular tag that screams B cell, is the expression of the B cell receptor, or BCR. This is a membrane-bound immunoglobulin, essentially an antibody molecule that directly binds to soluble or cell surface antigens.
The BCR is a surface antibody.
Each B cell is literally covered with a dense coat, carrying between 150,000 to 300,000 identical surface antibody molecules waiting to snag a matching antigen.
Covered in sensors.
Yeah.
And their ability to bind antigen isn't static. It's a dynamic, evolving process. B cells can actually improve their binding strength to an antigen through a process called somatic hypermutation.
Fine-tuning the antibody fit, making it stickier.
Fine-tuning their antibodies for even greater affinity. And they can class switch, generating different functional classes of antibodies like IgG, IgA, or IgE, each with distinct roles in immune defense.
Changing the antibody's function without changing its target. We'll explore these sophisticated processes in much more detail in a later chapter.
Looking forward to that. What's also incredibly unique about activated B cells is that they are the only non-myeloid cells that can act as professional APCs.
That's a really important point. They wear two hats, antibody producer and antigen presenter.
They efficiently internalize antigen via their BCR, process it into peptide fragments, and then, crucially, present those fragments on their surface as peptide MHC complexes to T cells.
Showing the T cells what they found. This allows them to receive that critical help from T lymphocytes.
And this direct interaction, receiving crucial T cell help, typically in the form of cytokines, is what truly induces their differentiation into antibody-producing plasma cells and long-lived memory cells.
That T cell help is often essential for a strong, long-lasting antibody response.
Plasma cells are the ultimate effector cells for antibody secretion. When a B cell transforms into a plasma cell, it undergoes a dramatic morphological change.
Think of it as a dedicated factory.
It loses its surface immunoglobulin expression and becomes a highly specialized antibody-secreting factory, capable of churning out hundreds to over 1,000 antibody molecules per second.
An incredible rate of production, just pumping out antibodies.
While some plasma cells can reside for years in the bone marrow, providing sustained antibody protection, others are relatively short-lived, dying within 1 or 2 weeks.
Short-term bursts and long-term suppliers. Importantly, these highly specialized cells have lost the capacity to divide. Their job is purely secretion.
Now let's move to T lymphocytes. Their T comes from their crucial maturation site, the thymus.
T for thymus. Makes sense.
Like B cells, T cells express a unique antigen-binding receptor called the T cell receptor, or TCR. But here's a fundamental and absolutely crucial difference from B cells.
A really key distinction.
TCRs only recognize processed pieces of antigen, typically small peptide fragments, that are bound to specialized cell membrane proteins called major histocompatibility complex, or MHC molecules.
They don't see whole antigens like B cells do. They need it presented on an MHC platter. This MHC restriction is a cornerstone of adaptive immunity.
MHC molecules are a family of highly diverse glycoproteins found on cell membranes. They were initially identified because they are the primary cause of rejection in transplanted tissues, hence histocompatibility.
Right, the molecules that determine tissue matching.
There are two main classes. MHC class I molecules are expressed by nearly all nucleated cells in your body.
Almost every cell has MHC class I.
Their job is to present peptides derived from internal proteins, whether they are normal self-proteins or foreign proteins, from, say, a virus replicating inside the cell.
Displaying what's going on inside the cell.
MHC class II molecules, on the other hand, are primarily expressed by professional APCs, like those macrophages and dendritic cells we discussed.
Restricted to the professional presenters.
They specialize in presenting peptides derived from external sources, such as engulfed bacteria. And here's a critical point. Helper T cells only recognize peptides presented on MHC class II molecules.
MHC class II is for helper T cells. MHC class I is for cytotoxic T cells. That's the rule.
This brings us to the two major types of T cells, which are distinguished by their co-receptor glycoproteins, CD4 or CD8.
The key identifiers.
T helper, or Th, cells predominantly display the CD4 co-receptor and are specialized to recognize antigen in complex with MHC class II molecules.
CD4 pairs with MHC class II.
Cytotoxic T, or TC, cells, in contrast, display the CD8 co-receptor and are designed to recognize antigen in complex with MHC class I molecules.
And CD8 pairs with MHC class I.
In healthy individuals, the CD4 to CD8 ratio in peripheral blood is approximately 2.1.
More helpers than killers, generally.
And a significant change in this ratio can be a strong indicator of various diseases, ranging from HIV infection, which targets CD4 plus T cells, to autoimmune disorders and chronic inflammation. It's a key diagnostic metric.
Doctors monitor that ratio closely.
Naive CD8 plus T cells, once they mature in the thymus, are constantly patrolling, browsing the surfaces of APCs in lymphoid organs.
Looking for trouble presented on MHC class I.
If their TCR binds to an MHC class I peptide complex that presents a foreign antigen, say, from a virus-infected cell, they become activated.
They find that target.
This activation triggers rapid proliferation and differentiation into cytotoxic T lymphocytes, or CTLs, the killer T cell. CTLs have an absolutely vital function. They monitor and directly eliminate cells that display non-self antigen complexed with MHC class I, essentially acting as the immune system's assassins for virus-infected cells, tumor cells, or foreign graft cells.
Taking out compromised host cells, it's important to note that for optimal activation and to become fully potent killers, these naive CD8 plus T cells often also need help from activated CD4 plus T cells.
So even the killers need some help sometimes.
Yeah, that helper signal licenses them to kill effectively.
Naive CD4 plus T cells also diligently browse the surfaces of professional APCs. Upon recognizing their specific MHC class II I peptide complex, they too become activated, proliferate extensively,
The helper cells find their cue on MHC class II.
and then differentiate into various distinct effector T cell subsets, each with a specialized role.
Not just one type of helper cell.
For instance, Th1 and Th17 cells primarily regulate responses to intracellular pathogens, like viruses or certain bacteria that hide inside host cells.
Helping fight invaders in ant cells.
In contrast, Th2 and TFH cells are crucial for regulating responses to extracellular pathogens, such as bacteria that remain outside host cells or multicellular parasitic worms.
Directing the fight against things outside cells, like coordinating antibody responses.
Each of these CD4 plus T cell subtypes produces a unique cocktail of different cytokines, which are signaling molecules that help activate other key immune cells, including B cells, CD8 plus T cells, macrophages, and various innate immune cells.
They're the conductors of the immune orchestra, using cytokines as their signals. The dominant subtype that emerges in an immune response depends heavily on the specific type of pathogen encountered, showcasing the finely tuned adaptability of the immune system.
And then we have a truly fascinating and critical population, regulatory T cells, or TREGs.
The peacekeepers of the immune system.
These cells possess the unique and vital capacity to inhibit immune responses. There are two main types. Natural TREGs, which develop in the thymus from cells that initially bind self-proteins with high affinity.
A fail-safe mechanism to prevent autoimmunity right from the start.
These are crucial for preventing autoimmunity by weeding out self-reactive T cells. The second types are induced TREGs, which can be generated at sites of inflammation or infection in the periphery.
Developed on the spot where needed to calm things down.
They are identified by their surface markers, CD4 plus and CD25 plus TREG, and crucially by the internal transcription factor, FOXP3.
FOXP3 is the master switch for TREGs.
TREGs are vital for quelling potentially autoreactive responses, preventing the immune system from attacking healthy cell tissue, and also for limiting normal T cell responses to pathogens, ensuring that the immune system doesn't overreact and cause excessive tissue damage after an infection.
putting the brakes on, absolutely essential for maintaining balance. What's more, research is still uncovering even greater diversity within the CD4 plus and TREG subpopulations, hinting at an even more nuanced regulatory network.
Then there are NKT cells, which are truly fascinating because they blur the lines, sharing characteristics with both T cells and natural killer NK cells.
A real hybrid cell type, bridging innate and adaptive immunity.
Effectively bridging the innate and adaptive immune systems. They possess TCRs, and some even express CD4, but their TCR repertoire is significantly less diverse than that of typical T cells.
They have T cell receptors, but not as varied.
Crucially, unlike conventional T cells that recognize protein peptides, NKT cells recognize lipids and glycolipids presented by specialized CD1 molecules, which are structurally related to MHC proteins.
They see fats, not proteins, presented on CD1, not MHC. Very unusual.
Adding to their hybrid nature, they also express receptors classically associated with innate immune cells, like NK receptors.
Borrowing features from the NK cell side, too.
When activated, NKT cells are dual-threat powerhouses. They can release cytotoxic granules to directly kill target cells, much like cytotoxic T cells or NK cells.
They can kill target cells directly.
But they also rapidly release large quantities of cytokines, which are signaling molecules that can have both pro-inflammatory enhancing and anti-inflammatory suppressing effects on immune responses.
And they release a flood of cytokines that can push the response in different directions. This makes them highly versatile modulators of immunity.
They are implicated in various human diseases, including asthma, and are actively being researched for their potential roles in inhibiting autoimmunity and even certain cancers.
Their precise functions in human health and disease remain an active and exciting area of investigation. A lot of potential there.
And finally, among the lymphoid lineage, we have the innate lymphoid cells, or ILCs.
A relatively newer family on the scene, but very important.
These cells are derived from common lymphoid progenitors, just like T and B cells. But here's the key distinction, the defining characteristic that separates them. They do not express antigen-specific receptors like the highly diverse TCRs or BCRs.
No unique rearranged receptors. They respond to more general danger signals.
This means they respond to broader, more conserved danger signals rather than specific pathogen peptides.
Part of the innate system, but with lymphoid origins.
Currently, ILCs are subdivided into three main groups: ILC1, ILC2, and ILC3. They are primarily distinguished by the unique cocktails of cytokines they secrete.
Groups by the signals they send out.
And what's remarkable is that these cytokine profiles precisely mirror those produced by distinct helper T cell subsets, as elegantly shown in Table 2.5 of your material.
Like innate versions of Th1, Th2, and Th17 cells, this suggests they act as rapid innate counterparts to T helper cells.
Many ILCs provide an immediate first line of defense against pathogens, particularly in mucosal tissues like the gut and respiratory tract, and in the skin, where pathogens first attempt to breach our defenses.
Stationed at the barriers for quick responses.
And among the ILCs, the best studied and perhaps the founding members of this category are natural killer, or NK cells.
The classic innate cytotoxic cell, often classified within ILC1 or as their own lineage.
They typically constitute 5–10% of human peripheral blood lymphocytes, and are identified by their expression of the NK1.1 surface protein, among others. These are truly efficient cell killers, employing two primary strategies to eliminate threats.
Very good at killing certain targets.
Their first killing strategy is unique. They attack cells that lack MHC class I molecules on their surface.
The missing self hypothesis.
Some viruses, for example, have evolved clever ways to evade cytotoxic T cells by forcing the infected host cell to downregulate its MHC class I expression, essentially hiding from T cell surveillance.
Trying to become invisible to CTLs.
NK cells, however, have inhibitory receptors that recognize MHC class I. When these receptors aren't engaged, the NK cell senses something is amiss, a missing cell signal, and releases its cytotoxic granules, inducing cell death in the target cell.
If the don't kill me signal, MHCI, is missing, the NK cell attacks. Clever counter strategy.
Our second strategy is called antibody-dependent cell cytotoxicity, or ADCC.
Using antibodies to find their targets.
NK cells express FC receptors, or FDRs, which are receptors for the constant region of antibodies. This allows them to arm themselves with specific antibodies, say, antibodies coating viral proteins on an infected cell surface.
They grab onto antibodies that have already tagged a cell.
Once the antibody brings the NK cell into close proximity with the target cell, the NK cell releases its potent granules, directly inducing cell death.
It's a powerful mechanism that links innate NK cell and adaptive antibody immunity. Our understanding of the entire ILC family, especially beyond NK cells, is still in its infancy, with new insights continually emerging and redefining their roles.
So we've now met the truly remarkable cellular cast of the immune system, from the stem cell that gives rise to all of them, to the specialized granulocytes, macrophages, dendritic cells, B cells, T cells, and ILCs.
An incredible diversity of players.
Now let's explore the crucial stages where these cells develop, mature, and where they ultimately launch their incredible defense. The organs and microenvironments of immunity.
Right, the anatomical context. Where does all this happen?
We begin with the primary lymphoid organs.
The schools and nurseries.
These are defined as the specialized sites where immune cells develop from their immature precursors undergoing crucial maturation and selection processes. The bone marrow is undoubtedly a central hub in this process.
Back to the bone marrow.
It's the unique environment where hematopoietic stem cells, those incredible HSCs we started with, reside in highly specialized microenvironments or niches.
Those supportive neighborhoods again.
These niches are lined by a complex network of supportive stromal cells that precisely regulate HSC survival, proliferation, differentiation, and even their ability to traffic out into the bloodstream when needed.
Controlling the whole HSC life cycle. What's truly remarkable is that the bone marrow remains the primary site of hematopoiesis throughout adult life, constantly churning out new blood and immune cells.
The lifelong factory.
It supports the maturation of all erythroid, myeloid, and in humans and mice, B lymphocytes.
Birthplace and school for many cell types. What's more, HSCs possess a remarkable natural capacity to recirculate between the bone marrow and other tissues.
They don't just stay put.
This natural mobility, unexpectedly, has significantly simplified complex medical procedures, like bone marrow transplantation, as we can now harvest HSCs from peripheral blood, not just directly from the marrow.
Yeah, mobilizing HSCs into the blood for collection is now standard practice, much easier on the donor.
This naturally leads us to Clinical Focus Box 2.2, which delves deeply into the clinical uses and immense potential of stem cells.
The therapeutic angle. The overarching promise here is nothing short of regenerating diseased, damaged, or defective tissue, fundamentally transforming medicine.
Huge potential.
The utility of stem cells comes directly from their dual, powerful properties. Their unparalleled capacity to give rise to diverse, differentiated cells, and their unique self-renewing ability.
Make specialists and make more of themselves.
Meaning each division of a stem cell creates at least one more stem cell, ensuring the pool is maintained.
They're broadly classified by their developmental potential. At the pinnacle are pluripotent stem cells, like those found in a fertilized egg or early embryonic stem cell,
The ultimate stem cells.
which possess the astonishing capability to give rise to an entire organism. The development of human embryonic stem cells, and more recently, induced pluripotent stem cells, iPSCs,
iPSCs were a massive breakthrough, turning adult cells back into stem cells.
which are differentiated. Adult cells reprogrammed back into a pluripotent state holds enormous therapeutic potential. Imagine generating replacement tissues like healthy skin for burn patients, functional heart muscle cells to repair damaged hearts, pancreatic beta cells for diabetes, or even new neurons for devastating neurodegenerative diseases like Parkinson's or Alzheimer's.
The dream of regenerative medicine. Still many hurdles, but incredible promise.
Beyond this pluripotent promise, HSC transplantation is already a vital and established therapy.
Used routinely in clinics today.
It's extensively used to provide a functional immune system for individuals born with severe genetic immunodeficiencies, such as severe combined immunodeficiency, or SCID, often called bubble boy disease.
Giving them a working immune system. Life-saving.
It can replace a defective hematopoietic system, literally curing life-threatening non-malignant genetic disorders like sickle cell anemia or thalassemia.
Replacing the faulty blood factory.
And crucially, it restores the entire hematopoietic system of cancer patients after they've undergone intensive chemotherapy or radiation that unfortunately destroys their own bone marrow, particularly for leukemias and lymphomas.
Rebuilding the system after wiping it out to kill the cancer. Essential for many cancer therapies.
The regenerative power of HSCs is truly mind-boggling. Think about it. Experiments in mice have demonstrated that as few as one single HSC can completely restore the entire erythroid population and the entire immune system in a lethally irradiated recipient.
One cell. It's almost unbelievable.
In humans, even a relatively small quantity, just 10% of a donor's total bone marrow volume, can be enough to completely restore the recipient's hematopoietic system.
Shows how potent these cells are.
And the ability to cryopreserve or bank these HSCs, often from a patient's own body before aggressive cancer treatment, is a revolutionary step, allowing for later autologous use without the risks of rejection.
Banking your own stem cells for later use, very important strategy.
However, despite their incredible promise,
Yeah.
transplantation isn't without its significant challenges, especially for allogeneic transplants, those from an unrelated donor.
Right, using someone else's cells brings immune complications.
While autologous, using one's own cells, or syngeneic from an identical twin, transplants have minimal complications, allogeneic transplants face the significant hurdle of immune rejection.
Host versus graft rejection.
Not only can the host's immune system reject the transplanted tissue, but tragically, the donor lymphocytes contained within the graft can attack the recipient's healthy tissues,
And the even more dangerous graft versus host disease, or GVHD.
leading to a severe and life-threatening condition called graft versus host disease, or GVHD. This necessitates powerful immunosuppressive drugs, which unfortunately carry their own risks, including increased susceptibility to infections and the development of secondary tumors.
A difficult balancing act. The best outcomes are, understandably, achieved when there is genetic identity or a very close match between the donor and recipient, minimizing these adverse immune reactions.
Thankfully, our medical understanding has led to significant advances in sourcing HSCs. While direct bone marrow aspiration from the hip used to be the primary and most invasive option,
Painful and requires anesthesia.
peripheral blood and umbilical cord blood are now common and often preferred alternative sources. Peripheral blood harvesting avoids invasive surgical procedures. Specific agents are used to induce HSCs to circulate from the bone marrow into the bloodstream,
Mobilizing them out of the marrow.
where they can then be collected via leukapheresis, a process that separates HSCs from other blood components.
Like a specialized blood donation.
Umbilical cord blood is also an increasingly attractive source due to its relatively high frequency of HSCs and the fact that it's derived from normally discarded placental tissue.
A valuable resource that used to be thrown away.
However, it's worth noting that engraftment, the successful integration and growth of the transplanted cells, can sometimes be less reliable with cord blood, often due to lower cell numbers.
It can take longer to rebuild the system with cord blood sometimes. Pros and cons to each source.
And beyond cancer treatment, autologous HSC transplantation is proving invaluable for advanced gene therapy.
Fixing genetic diseases at the source.
The introduction of a corrected gene into a patient's own HSCs, such as the ADA gene for SCID, has shown remarkable success in restoring immune function.
Taking the patient's cells out, fixing the gene, putting them back in.
While early methods using retroviral vectors sometimes led to unforeseen complications like leukemia due to random gene integration,
Yeah, that was a serious setback where the virus inserted caused cancer.
the promise of new, more precise gene editing techniques, such as CRISPR-Cas9, holds immense potential for safer and far more efficient gene delivery into HSCs, offering hope for a wide range of genetic diseases in the future.
CRISPR offers the potential for much more targeted, safer gene correction. Very exciting field.
Diving back into the bone marrow microenvironment itself, Figure 210 in your material gives us a vivid picture. It's a complex, sponge-like medullary cavity nestled within the bones.
The inside of the bone.
But it's not just a passive space. Within it, HSCs reside in very distinct, highly specialized niches. We have the endostele niche, which lines the bone surface and is closely associated with osteoblasts, the cells that form bone, and B lymphocyte progenitors.
Close to the bone itself seems important for B cells.
Then there's the perivascular niche, which lines the central blood vessels. This niche specifically supports quiescent, long-lived HSCs, providing a nurturing environment thanks to close interactions with perivascular cells and endothelial cells.
Near the blood vessels seems to be where the long-term, quiet HSCs hang out.
These stromal cells, a diverse cast, including endothelial cells, perivascular cells, even sympathetic nerves, macrophages, and osteoblasts, all work in concert to precisely regulate every aspect of HSC function.
The whole neighborhood supports and controls the HSCs.
Their quiescence, their proliferation, their trafficking within and out of the bone marrow, and their differentiation pathways. While specific niches for myeloid development are still being elucidated, It's clear that B lymphocytes complete the vast majority of their maturation right here in the bone marrow,
B cells mostly grow up in the marrow.
with their progenitor specifically found in the endothelial niche. T lymphocyte progenitors, however, have a different journey. They leave the bone marrow at a very immature stage to complete their crucial development in another primary lymphoid organ, the thymus.
T cells need to go elsewhere for their education.
And it's also worth noting that the bone marrow isn't just a site for cellular development. It's also a place where many mature myeloid and lymphoid cells, particularly antibody-secreting plasma cells, and even some memory T cells, can return and reside long-term.
A home base for mature cells, too, especially those long-lived plasma cells churning out antibodies, continuing their essential functions.
And speaking of the thymus, this truly is the T cell finishing school.
The elite academy for T cells.
T cell development, with all its critical educational steps, isn't complete until these cells undergo rigorous selection processes within this specialized, highly controlled environment.
It's a tough school, high failure rate.
What's absolutely wild is that the profound importance of the thymus wasn't even recognized until the early 1960s. For centuries, it was largely dismissed, thought to be a mere vestigial organ, a cellular graveyard, or even remarkably something detrimental.
Yeah, its function was a real mystery for a long time. Hard to believe now.
This brings us to classic experiment box 2-3, and the truly groundbreaking work of J.F.A.P. Miller in 1961.
Miller's pivotal experiments.
He published observations in the prestigious Lancet Medical Journal that completely overturned prevailing notions. He showed that when the thymus was surgically removed from very young mice, a procedure known as a thymectomy, those mice became profoundly susceptible to infections, were unable to reject foreign skin grafts, and tragically died prematurely.
Removing the thymus had catastrophic immune consequences.
It became clear they were missing a crucial cell type, which was later identified and named T cells for thymus-derived.
The proof is in the pudding. No thymus, no functional T cells.
This directly challenged decades of scientific dogma and proved unequivocally that the thymus was a vital immunological organ, actively producing functional immune cells essential for adaptive immunity.
A paradigm shift in immunology, thanks to Miller's work.
Miller, like any groundbreaking scientist, faced significant criticisms initially, but he meticulously addressed every challenge experimentally, reproducing his data in different mouse strains and even in germ-free conditions.
He rigorously defended his findings against skepticism.
Ultimately vindicating his work and solidifying the thymus's place as the all-important site for T lymphocyte maturation. And then, surprisingly, in 2006, Hans Reimer Rodewald and colleagues made a fascinating discovery: a second thymus in mice.
A cervical thymus up in the neck? Who knew?
They found cervical thymic tissue in the neck, smaller but functionally contributing to the mature T cell repertoire. This discovery has even raised intriguing questions about the completeness of some past thymectomy studies and their interpretations, hinting at further evolutionary complexities.
Nature always has surprises. Maybe there's redundancy or subtle differences.
The T cell maturation journey in the thymus, vividly illustrated in Figure 211, is a complex, multi-stage process akin to an intensive boot camp for immune cells.
Yeah, they go through a real ordeal.
Immature T cell precursors, known as thymocytes, originate in the bone marrow and travel via the bloodstream to the thymus.
Making the trip from marrow to thymus.
Once inside, they embark on a precisely choreographed migration, passing through distinct microenvironments, each populated by specialized epithelial cell subtypes that provide critical signals.
Moving through different zones, getting different lessons.
They enter at the corticomedullary junction, the interface between the outer cortex and inner medulla. They then move to the subcapsular cortex, just beneath the thymus's outer capsule, where the youngest, double negative DN thymocytes, meaning they express neither CD4 nor CD8, proliferate extensively.
Starting out DN and multiplying.
As they move deeper into the cortex, thymocytes begin to express both CD4 and CD8, becoming double positive DP cells.
Turning on both CD4 and CD8.
It's here in the cortex that they undergo positive selection. This is a crucial screening step where they interact with cortical thymic epithelial cells, or CTECs.
First big test. Can you recognize MHC at all?
Only those thymocytes whose newly rearranged TCRs can bind to self-MHC peptide complexes with intermediate affinity receive survival signals and are positively selected.
Not too strong, not too weak. Goldilocks binding.
Those that bind too weakly, the majority, actually die by death by neglect and are essentially eliminated from the repertoire.
Most fail this test. If you can't recognize self-MHC, you're useless.
But the rigorous screening isn't over. Next comes negative selection, which is vital for preventing autoimmunity.
Second big test. Do you recognize self too strongly?
This can occur in any microenvironment, but is especially crucial in the medulla, with specialized medullary thymic epithelial cells, MTX. Here, thymocytes, whose TCRs bind to self-MHC peptide complexes with too high affinity, are induced to die through apoptosis.
Eliminating the potentially self-reactive cells. Critical for tolerance.
This eliminates potentially autoreactive T cells that might attack healthy self-tissue. The attrition rate during this entire maturation process is incredibly high. Over 95% of all thymocytes that enter die in transit.
95%, a testament to the stringent quality control necessary to produce a functional yet self-tolerant T-cell repertoire.
As they progress, thymocytes transition from double negative, DN, expressing neither CD4 nor CD8, to double positive DP, expressing both CD4 and CD8 in the cortex.
DN to DP.
Then, as they mature further and pass through selection, they lose one CD antigen, becoming single positive SP cells, either CD4 plus for helper T cells or CD8 plus for cytotoxic T cells.
Then DP to SP, committing to being either a helper or a killer.
Effectively determining their future functional role. These mature SP cells then exit the thymus, primarily via the corticomedullary junction blood vessels, as recent thymic immigrants, ready to finalize their maturation and contribute to the adaptive immune response in the periphery.
Graduated, but still need a bit of real-world experience.
We've now covered the birthplaces and finishing schools for immune cells. But how do these specialized cells actually get around to where they're needed?
Right, the logistics. That's where our two major circulatory systems come in. The blood and the lymphatics, which work in a beautiful coordinated dance to patrol and connect the entire body.
Blood vessels, quite simply, provide rapid access to virtually every organ and tissue in your body.
The highways of the body.
Arteries, with their thick muscular walls, efficiently pump blood away from the heart, while veins, with thinner walls and one-way valves, return it. This is a system of rapid transit, ensuring oxygen, nutrients, and immune cells can quickly reach distant sites.
Fast delivery system.
Crucially, the endothelial cells lining these blood vessels play an active role, cooperating with innate immune cells to specifically recruit white blood cells to infected or inflamed tissues,
They're not just passive pipes. They actively control who gets out.
allowing them to squeeze between endothelial cells and follow molecular breadcrumbs called chemokine gradients to the precise site of action.
Following the scent to the site of inflammation.
The lymphatic system, on the other hand, is a distinct and equally vital network. It's a vast system of vessels filled with protein-rich lymph, a fluid derived from blood plasma that seeps out of capillaries.
The other circulatory system. Kind of the drainage and surveillance network.
Uniquely, only white blood cells lymphocytes and some APCs have regular access to the system for immune surveillance.
Red blood cells generally stay out of the lymphatics.
The lymphatic system serves two absolutely critical functions. First, it's the primary route for activated immune cells and antigens to travel from sites of infection or tissue injury to the secondary lymphoid organs, like the lymph nodes,
Transporting intel and activated cells to headquarters.
where they meet and activate lymphocytes. Most secondary lymphoid tissues, with the notable exception of the spleen, which is primarily blood-served, are strategically situated along these lymphatic vessels.
Lymph nodes are dotted along the lymphatic highways.
And its second equally vital function is fluid return. Your blood capillaries are leaky, and about 2.9 liters or more of interstitial fluid that seeps out of your blood vessels and surrounds your cells accumulates in your tissues every 24 hours.
A lot of fluid leaks out.
The lymphatic system is essential for collecting this fluid and returning it back to the circulation. Without this constant drainage, your tissues would swell dramatically, leading to edema, or in severe cases, lymphedema.
Prevents swelling, essential for fluid balance, which can be life-threatening.
Lymphatic vessels have thinner, more porous walls than blood vessels, composed of a single layer of loosely opposed endothelial cells, allowing for the easy entry of fluids, cells, and even large antigen-antibody complexes.
Easier access for immune surveillance cargo.
The flow within lymphatic vessels is slow and low pressure, relying on one-way valves and the action of surrounding muscles. Physical activity, in fact, enhances lymph circulation.
Moving your muscles helps pump the lymph along.
The largest lymphatic vessel, the thoracic duct, collects lymph from most of the body and empties into the left subclavian vein, while the right lymphatic duct drains the right arm and head into the right subclavian vein, eventually returning everything to the blood.
Draining back into the bloodstream near the heart.
And guiding all this complex cellular movement are chemokines.
The molecular GPS system.
Think of these small molecules as the precise GPS signals for immune cells. They are a family of chemoattractants secreted by a wide variety of cells, from epithelial cells, forming our barriers, to stromal cells providing structural support, to professional APCs, and even lymphocytes and granulocytes themselves.
Lots of cells can produce these signals.
Immune cells express diverse chemokine receptors on their surface, allowing them to sense these chemical gradients and migrate precisely towards the source of chemokine production.
Following the chemokine trail, this ensures that the right cells get to exactly where they're needed most, whether it's an infection site or a specific zone within a lymph node. This system is critical for directing the finely choreographed immune response.
Now let's explore the secondary lymphoid organs. These are the actual command centers, the sites where mature antigen-specific lymphocytes first encounter their specific antigen.
Where the adaptive immune response really kicks off, the meeting places.
Finally get activated and begin the crucial process of differentiating into those potent effector cells and long-lived memory cells. These include the highly organized encapsulated structures like lymph nodes and the spleen,
The major well-defined organs.
as well as the more loosely organized lymphoid tissues found in our barrier organs, such as the skin and mucosal membranes.
And the defenses built into our barriers.
Despite their varied structures, all secondary lymphoid structures share several key features. They all possess anatomically distinct regions specifically dedicated to T-cell activity and B-cell activity, ensuring optimal interactions.
T-cell zones and B-cell zones keeps things organized.
And critically, they all have the capacity to generate lymphoid follicles, which are highly organized microenvironments essential for the development and selection of B cells that will ultimately produce high-affinity antibodies.
Follicles are the B-cell hubs, especially for making great antibodies.
The lymph node is truly a regional hub, fully committed to regulating the immune response in a specific anatomical area, unlike the spleen, which also has a major role in managing red blood cell flow.
Lymph nodes drain specific territories of the body, very localized surveillance.
Lymph nodes are encapsulated, bean-shaped structures, typically found clustered in areas like your neck, armpits, and groin. They are densely packed with a dynamic network of stromal cells, lymphocytes, macrophages, and dendritic cells,
Busy intersections of immune cells.
and are intricately connected to both the blood and lymphatic vessels. They are the first organized lymphoid structures to encounter antigens that originate from tissue spaces, making them ideal for providing the precise microenvironments for antigen lymphocyte encounters.
Sampling the fluid draining from nearby tissues, perfect place to catch trouble early.
A lymph node is beautifully organized into three concentric regions, like layers of an onion.
A defined architecture.
The outermost layer is the cortex, which is rich in B cells, macrophages, and specialized follicular dendritic cells, FDCs, found within distinct B cell follicles.
Cortex is B-cell zone with follicles.
Beneath that is the paracortex, which is largely populated by T lymphocytes and the crucial dendritic cells that have migrated in from the peripheral tissues, carrying captured antigens.
Paracortex D-cell zone, where the DCs arrive.
And finally, the innermost region is the medulla, which is more sparsely populated but contains actively secreting plasma cells that churn out antibodies, and it's also the site where activated immune cells exit the lymph node via efferent lymphatics.
Medulla exit zone, and where some plasma cells hang out.
So how does an antigen actually get into this bustling hub?
Delivery mechanisms.
Antigen travels from an infected tissue, perhaps a cut finger, via afferent lymphatic vessels, which pierce the lymph node capsule and empty into the subcapsular sinus.
Arriving via the incoming lymph.
This antigen can be particulate, like a whole bacterium, or it can already be processed and presented on the surface of migrating APCs, particularly dendritic cells that have picked up the pathogen in the tissues.
Coming in various forms.
Once inside, the antigen is efficiently trapped by resident APCs in the subcapsular sinus and cortex, which then pass it on to B cells in the follicles, or presented by resident DCs directly to the T cells in the T-cell-rich paracortex.
Filtered and presented to the right audience. It's a very efficient system for antigen delivery.
For T cells in the lymph node, it's a process of rapid yet meticulous browsing. A single naive T lymphocyte takes an astonishing 16 to 24 hours to thoroughly browse the vast array of MHC peptide combinations presented by APCs in just one lymph node.
Takes a whole day just to check one node. Shows how thorough the surveillance is.
Naive lymphocytes constantly recirculate, entering the lymph node from the bloodstream via specialized high endothelial venules, or HEVs,
Entering from the blood through special doorways, the HEVs.
which are lined by tall endothelial cells that facilitate their entry. Once inside the paracortex, the designated T cell zone, they don't just wander aimlessly.
They follow specific paths.
They actively browse APCs that are strategically positioned on a sophisticated network of fibroblastic reticular cells, or FRCs, which form a highly organized conduit system, FRCC.
Like highways made of stromal cells guiding the T cells.
This physical network, combined with specific adhesion molecules and chemokine gradients produced by the FRCs, guides T cell movements, dramatically enhancing the probability of a specific T cell meeting its cognate antigen.
Maximizing the chances of that rare T cell finding its one specific match among millions of possibilities. The one MHC peptide complex it's been waiting to recognize.
What happens next is all about that recognition. If a T cell doesn't find its specific MHC peptide match within a certain time frame, it simply exits via the efferent lymphatics in the medulla.
No match. Okay, moving on to the next node.
Continuing its journey to browse other lymph nodes, endlessly patrolling. But if it does bind to its specific antigen, it stops migrating abruptly.
Found it. Stop the search.
It then resides in the lymph node for several days, undergoing rapid proliferation to expand its numbers, and crucially differentiates into powerful effector cells.
Setting up shop to multiply and arm itself.
CD8 plus T cells become cytotoxic T lymphocytes, CTLs, gaining their full killing ability, while CD4 plus T helper cells differentiate into various effector types that go on to activate macrophages, help other CD8 plus T cells become fully armed, and crucially, provide essential help to B cells.
Becoming killers or specialized helpers.
B cells in the lymph node also perform a highly specific and coordinated dance. For optimal B cell activation, two signals are generally required. Initial antigen engagement by the B cell receptor.
Signal 1, antigen binding.
And then crucially, direct contact with an activated CD4 plus T helper cell.
Signal 2, T cell help.
Naive B cells also circulate, entering the lymph node via HEVs, but they're specifically drawn to the lymph node follicles, not the paracortex, by distinct signals and chemokine gradients.
Heading straight for the B cell zones.
They initially utilized the FRC conduit system for rapid transit, but then shift to specialized tracks generated by follicular dendritic cells, FDCs.
Switching tracks to follow the FDC network in the follicle.
Which, as we mentioned, specialize in presenting unprocessed particulate antigen directly to B cells.
Remember, FDCs show whole antigens to B cells.
Unlike T cells, B cells have the remarkable ability to recognize free, unprocessed antigen. Small, soluble antigens can enter the follicles directly, but larger antigens, like whole bacteria, are efficiently relayed by specialized subcapsular macrophages or even non-antigen-specific B cells to the FDCs within the follicle.
Getting the antigen delivered to the B cells in the right form?
If a B cell's BCR binds its specific antigen, it becomes partially activated. It then internalizes that antigen, processes it, and loads it onto MHC class II molecules.
Grabs the antigen, eats it, processes it,
Ready to present it as a peptide MHC complex to those helper T cells.
and puts pieces on its own MHC class II to ask for help, becoming an APC itself temporarily.
This partially activated B cell then migrates from the follicle to the T cell-rich paracortex, actively seeking an activated CD4 plus T helper cell that recognizes the same antigen.
Moving to the T cell zone to find its partner.
If the T cell recognizes the B cell's presented MHC peptide complex, they form a stable, sustained contact that can last for hours. This critical interaction delivers the T cell help needed,
The T cell confirms the B cell found something important.
triggering profound B cell proliferation and differentiation. Some activated B cells will differentiate directly into antibody-producing plasma cells, ready to secrete antibodies immediately.
Quick response plasma cells.
But others embark on a fascinating journey. They re-enter the follicle to establish a highly dynamic structure called the germinal center,
Back into the follicle to form a germinal center.
effectively forming a secondary follicle.
This is where the really high-quality antibody response is generated.
And within these germinal centers, a complex process unfolds. B cells undergo rapid proliferation, hypermutation of their antibody genes, and a rigorous process of clonal selection to produce antibodies with incredibly high affinity for the antigen.
Intense evolution in miniature. Survival of the fittest antibody. It's like an intense evolutionary pressure cooker for antibodies.
From here, some of these highly efficient antibody-producing cells go to the medulla of the lymph node to release their potent antibodies directly into the bloodstream providing systemic protection.
Sending antibodies out into circulation.
Others exit the lymph node via the efferent lymphatics to become long-term residents in the bone marrow, continuing their critical antibody release for years.
Becoming those long-lived plasma cells in the marrow.
It's why when you have an infection, your lymph nodes often visibly swell and can be painful. It's a direct result of this intense cellular migration, proliferation, and formation of these bustling germinal centers.
All that activity makes the nodes swell up, which typically establish 4 to 7 days post-infection and remain active for 3 or more weeks.
And all these intricate interactions between T cells and professional APCs, and between activated T helper cells and B cells, ultimately lead to the generation of long-lived memory T and B cells.
The crucial outcome, lasting memory.
These vital cells don't just stay put. They can reside in secondary lymphoid tissues, constantly circulate throughout the body, or become specialized tissue-resident memory cells, strategically positioned in peripheral tissues to act as immediate first responders to any reinfection.
Memory cells patrol everywhere or stand guard in tissues. This area of understanding, the formation and maintenance of tissue-resident memory, is a very active and exciting area of research, with profound implications for vaccine design and sustained immunity.
Okay, lymph nodes covered. Let's move to the spleen, our bloodborne pathogen specialist.
Exactly. The spleen filters the blood, whereas lymph nodes filter the lymph.
Located in the upper left abdominal cavity, it plays an absolutely major and distinct role in mounting immune responses, specifically to antigens encountered in the bloodstream.
Dealing with systemic infections that get into the blood.
This makes it crucial for systemic infections, where pathogens are circulating throughout the blood. Unlike lymph nodes, the spleen is not supplied by lymphatics. Bloodborne antigens and lymphocytes enter directly via the splenic artery and process blood exits via the splenic vein.
All traffic is via the blood for the spleen.
It's a massive filter.
Yeah.
More recirculating lymphocytes pass daily through the spleen than through all lymph nodes combined, highlighting its central role in blood surveillance.
Huge volume of blood passes through, constantly checked by lymphocytes.
The spleen is a beautifully organized organ surrounded by a capsule and broadly divided into 2 main functional compartments, the red pulp and the white pulp, separated by a specialized marginal zone.
Red pulp and white pulp, distinct jobs.
The red pulp is a vast network of sinusoids, densely populated by red blood cells, macrophages, and lymphocytes. This is the primary site for the destruction of old or defective red blood cells.
The recycling center for old red blood cells. Macrophages here are constantly engulfing and recycling them, leaving telltale iron pigments.
Crucially, pathogens circulating in the blood often gain their first access to the immune cells in the white pulp from the red pulp.
Pathogens can get caught here and presented to the immune parts.
The white pulp is the true immune powerhouse of the spleen. It surrounds branches of the splenic artery, forming distinct cylindrical structures.
This is where the adaptive response happens in the spleen.
It contains organized B-cell follicles and a T lymphocyte-rich periarteriolar lymphoid sheath, or PALS.
PALS is the T-cell zone of the spleen surrounding the arteries. Follicles are nearby.
Just like in lymph nodes, germinal centers dynamically form here during an immune response to generate high-affinity antibodies. A sophisticated fibroblastic reticular network within the white pulp also helps guide the precise migration of T and B cells, ensuring optimal encounters.
Similar organizational principles as the lymph node, but adapted for blood filtering.
The marginal zone, or MZ, is a specialized cellular border, a dynamic interface between the bloodstream and the white pulp.
A unique transition zone.
It's populated by unique cell types, including specialized dendritic cells, macrophages, and particularly unique marginal zone B cells, or MZB cells.
MZB cells are special residents of this zone.
These MZB cells are thought to be a crucial first line of defense against bloodborne pathogens. They are strategically positioned to rapidly trap antigens directly from the splenic artery.
Right at the entry point, ready to grab pathogens immediately.
What's fascinating about them is that they express both innate immune receptors, like toll-like receptors, TLRs, and unique B-cell receptors tailored to recognize conserved patterns on pathogens.
Sensing general danger signals and specific antigens. Quick responders.
This allows them to differentiate rapidly and secrete high levels of antibodies, often T-independent antibodies, providing quick, broad protection, though some can also receive T-cell help.
A rapid antibody response, sometimes without needing T-cells. It's worth noting there are some subtle differences in MZ B-cell populations between mice and humans, a nuance that researchers are still exploring.
The initiation of adaptive immune responses within the spleen is highly analogous to that in lymph nodes. Naive B cells encounter their specific antigens within the follicles. Naive CD8 plus and CD4 plus T cells meet their specific MAC peptide complexes presented by dendritic cells in the PLS, which is the T cell zone.
Same basic principles. B cells in follicles, T cells in the PLS meet DCs.
Activated CD4 plus T cells then provide essential help to both B cells, including those important MZ cells, and CD8 plus T cells, leading to the formation of those crucial germinal centers.
T cell help drives the response, forming germinal centers.
Ultimately, this leads to the generation of effector cells, as well as long-lived memory B and T cells and antibody-secreting plasma cells that can circulate throughout the body or reside in the bone marrow, providing sustained protection.
Same outcomes, effectors, memory, plasma cells.
The critical and irreplaceable role of the spleen is starkly highlighted by the implications of a splenectomy, the surgical removal of the spleen.
Losing the spleen has serious consequences.
Children who undergo this surgery are particularly vulnerable to a life-threatening condition called overwhelming post-splenectomy infection, or OPSI, especially from systemic bacterial infections caused by encapsulated bacteria like Streptococcus pneumonia, Neisseria meningitis, and Haemophilus influenzae.
Those encapsulated bacteria are usually cleared efficiently by the spleen. Without it, infections can be devastating, especially in kids.
While less severe in adults, splenectomy still significantly increases vulnerability to blood-borne bacterial infections, underscoring the spleen's vital role in clearing pathogens from the bloodstream.
Highlights its unique role in filtering blood. Beyond immunity, it also compromises iron metabolism and platelet storage, showcasing its multifaceted importance.
Okay. Beyond these highly organized encapsulated organs, we also have our barrier organs, which possess their own specialized, often more loosely organized, secondary lymphoid tissue.
The defense is embedded right at our surfaces.
Think about the sheer surface area. Our skin and mucosal membranes of the digestive, respiratory, and urogenital tracts collectively cover over 400 square meters, roughly the size of a basketball court.
A huge area exposed to the outside world.
These vast surfaces are, quite literally, the major entry points for the vast majority of pathogens we encounter daily.
Need strong, localized defenses.
These vulnerable surfaces are strategically defended by collections of organized lymphoid tissues, collectively known as mucosa-associated lymphoid tissue, or MALT.
MALT, the umbrella term.
This umbrella term includes specific regional variants like BALT, bronchus-associated lymphoid tissue, NALT, nasal-associated, GALT, gut-associated, and SALT, skin-associated.
Name for the location. GALT in the gut is probably the biggest.
The epithelial cell layers that form these barriers aren't just passive physical protection, they're active participants in immunity, constantly secreting cytokines, chemokines, and potent antimicrobial compounds.
The barrier cells themselves fight back.
Immune cells reside in deeper layers within these tissues, generating B cell follicles, where B cells tend to secrete a specific type of antibody called IgA.
IgA is the characteristic antibody at mucosal surfaces.
What's remarkable about IgA is its ability to cross epithelial barriers and directly interact with microbes right there in the lumen of the gut or respiratory tract, providing a direct, localized defense.
Secreted out onto the surface to neutralize threats before they even invade.
These barrier immune systems also play a profoundly crucial role in a delicate balancing act, maintaining tolerance to the vast and diverse commensal microbes that coexist harmlessly and often beneficially within us.
Distinguishing friend from foe. Tolerating the good bacteria while fighting the bad. Hugely important in the gut.
A prime example of GALT is the Peyer's patch, extensive lymphoid nodules found in the intestinal submucosa.
Peyer's patches are like specialized lymph nodes embedded in the gut wall.
Specialized epithelial cells, called M cells, in the epithelial layer of the Peyer's patch, are designed to actively convey antigens from the intestinal lumen to the underlying immune cells in the lamina propria.
M cells act like sample collectors, pulling antigens from the gut lumen inside.
This targeted antigen delivery triggers B cell follicle formation and IgA production, which then passes back into the intestinal lumen, forming a protective barrier.
Generating that local IgA response right where it's needed.
Other cells like resident macrophages, dendritic cells, and specialized intraepithelial lymphocytes also constantly sample antigens, working in concert with regulatory T cells to distinguish beneficial microbes from dangerous pathogens.
The whole team working to sample, respond appropriately, or tolerate.
Moreover, activated APCs and lymphocytes from the pairs patch can also travel to local lymph nodes, ensuring that if a systemic threat emerges, a broader immune response can be launched.
Reacting the local gut response to the systemic network.
Finally, we arrive at tertiary lymphoid tissues.
These are different, not preformed structures.
These are distinct from primary and secondary lymphoid organs because they are not preformed, anatomically defined structures. Instead, they are dynamic sites of active infection, inflammation, and intense immune activity that develop de novo in peripheral tissues.
They pop up where chronic inflammation or infection is happening.
Once lymphocytes are activated in the spleen or lymph nodes, they migrate out as effector cells and can return to these areas, like the lung, liver, brain, or skin, to fight the infection. They can also reside there long-term as specialized tissue-resident memory cells.
Activated cells go to the site of the battle, and memory cells can stay there on guard duty, providing immediate on-site protection.
What's truly remarkable, and a testament to the incredible plasticity of tissue anatomy, is that tertiary lymphoid tissues can spontaneously generate new, organized lymphoid microenvironments in response to chronic infection or persistent inflammation.
The tissue can actually build mini lymph node-like structures on demand.
Our sources give the fascinating example of the brain-forming reticular systems to guide lymphocytes during chronic toxoplasmosis infection, showing a level of dynamic organization previously thought to be exclusive to secondary lymphoid organs.
Building guidance structures even in the brain. Amazing adaptability. These organized aggregations of immune cells are particularly prominent in chronic infections and autoimmune diseases, highlighting the intimate and adaptable relationship between immune cells and the non-immune cells and structures of peripheral tissues.
This profound structural plasticity is further illuminated by Evolution Box 2.4, which details the fascinating anatomical variations in immune systems across vertebrates.
Taking the long view, how did this all evolve?
The adaptive immune system, with its defining ability to generate antigen-specific receptors, first appeared approximately 500 million years ago with the emergence of vertebrates.
A relatively recent invention in evolutionary terms tied to vertebrates.
Interestingly, older immune tissues generally tended to be retained as newer, more complex organs appeared through evolutionary time. For instance, all vertebrates, from fish to humans, possess some form of GALT,
Gut immunity seems ancient and essential.
indicating its ancient and fundamental importance in gut immunity. However, only jawed vertebrates possess well-developed thymi and spleens, highlighting a key evolutionary divergence. Yet despite these variations, all jawed vertebrates have populations of both B and T cells.
Thymus, spleen, B, and T cells seem to arrive with jawed vertebrates.
T cells were the first lymphocytes to express diverse antigen receptors, and their appearance is directly linked to the evolution of the thymus. All jawed vertebrates have a thymus.
T cells and the thymus go hand in hand evolutionarily.
What's truly fascinating is that even jawless vertebrates, like the ancient lamprey, possess thymic-like tissue, often called thymoid tissue, found in their gill regions.
A precursor to the thymus, even in jawless fish, suggests the pathway was starting early, suggesting an ancient precursor to the specialized T-cell maturation organ.
But here's another surprising evolutionary twist. The precise site of B-cell development isn't bound to one single organ.
B cells are more flexible about where they grow up.
In mammals like mice and humans, it's primarily the bone marrow, with further maturation in the spleen. But in cattle and sheep, it's the ileal pairs patch in the gut. In rabbits, it's gut-associated tissues like the appendix.
Different mammals use different primary B-cell organs.
And recent data suggests that the gut can actually act as a primary lymphoid organ for B cells in most vertebrates, showcasing remarkable functional convergence.
The gut seems to be a recurring theme for B-cell development across evolution.
And when we look at the evolution of secondary lymphoid tissues, we see a clear progression towards increased complexity. The spleen is considered the most ancient secondary lymphoid organ, present in many early vertebrates.
Spleen came first.
Lymph nodes, however, are the most recent immune adaptation, becoming most highly organized and abundant in mammals.
Lymph nodes are the newest addition, most developed in mammals.
But even within mammals, there's striking plasticity. Consider the remarkable example of pig lymph nodes. They are anatomically inverted.
Upside down lymph nodes in pigs, what?
The medulla, which is typically the exit zone, is on the outside. And the cortex, where antigen presentation and cell proliferation occur, is on the inside.
Completely reversed architecture.
Furthermore, lymphocytes exit pig lymph nodes via blood vessels, not efferent lymphatics.
They leave through the blood, not the lymph. The adaptive advantages of this inverted anatomy are still unknown, but it powerfully reminds us of the remarkable flexibility and diversity in biological structures and evolutionary processes, constantly adapting to environmental pressures.
So what does this all mean for you, the listener, and for our deeper understanding of immunity? We've journeyed today from the single, potent hematopoietic stem cell, that incredible progenitor,
In the very beginning.
through its mind-boggling differentiation pathways into the vast, diverse cellular army of granulocytes, macrophages, dendritic cells, B cells, T cells, and innate lymphoid cells.
Is that the whole cellular cast?
We've then meticulously explored the specialized primary organs, the bone marrow and thymus, where these cells are born and undergo their crucial maturation.
So where they go to school.
And finally, we've examined the secondary and even tertiary lymphoid tissues, which act as dynamic staging grounds where these cells organize, meet their specific antigens, and launch their finely tuned, life-saving immune responses.
And explored the command centers and battlefields.
What's truly fascinating here, and I hope we've conveyed this depth, is the sheer level of orchestration and coordination within this system.
It's also interconnected and regulated.
From microscopic chemokines, precisely guiding cell traffic, to the complex, intricate stromal cell networks in lymph nodes, and the spleen that ensure optimal encounters between incredibly rare specific lymphocytes and their cognate antigens.
Every detail, from molecules to tissues, is designed for efficiency.
Every single element, down to the last molecular detail, is designed with breathtaking efficiency. It's a testament to millions of years of evolutionary refinement, optimizing our very survival against an ever-present world of threats.
It really is. A beautiful biological machine honed by evolution.
This incredible complexity and adaptability raise an important and truly provocative question for you to consider. Given the constant, relentless evolution of pathogens and the surprising dynamic plasticity we've observed in immune system anatomy across species,
Thinking about the future.
what new immune cells or even entirely new organs might still be discovered within us? Are there silent cellular adaptations, perhaps forming in response to novel threats that we haven't yet identified?
Could there be more hidden layers we haven't found yet?
And how might a deeper understanding of this profound biological plasticity, the ability of our immune system to dynamically reshape itself, transform future medical interventions, allowing us to harness or even re-engineer our own defenses to combat diseases we can barely imagine today?
Can we learn to manipulate this plasticity for therapeutic benefit? Huge potential there.
So the next time you feel a swollen lymph node in your neck, remember, it's not just a bump. It's a living testament to this dynamic, constantly adapting blueprint of defense, silently at work inside you.